Hello, and thank you for attending today's webinar, Definitive QT Evaluation, Clinical and Operational Considerations, presented by Dr. Vince Clinical Research, Clario, and Fierce Biotech. My name is Janelle Karimoto, and I'll be your moderator. Before we begin, here are a few quick housekeeping notes. To learn more about our speakers, please visit the speaker bio window. Additional resources can be found in the handouts window. If you accidentally close the window, you can reopen it by clicking its name in the top navigation bar. This webinar is being recorded and will be available on demand within 24 hours. And finally, we will host a Q&A session after the presentations. Please submit your questions at any time throughout the webinar using the Submit Questions window. For context, when navigating the challenging QT landscape, sponsors must consider many factors when pursuing definitive QT assessments for NDA submissions. Fortunately, there are partners who can assist in this complex landscape and support the path to obtaining the required regulatory data. Many companies now leverage early QT assessments in their SAD trials, using this data with the hope of securing an FDA waiver. However, some companies may still be required to conduct a full, thorough QT study, which is significantly larger in scope and cost. This raises a common question: which pathway is right for your investigational product? Should you conduct a full, thorough QT study or rely on early QT assessments within your SAD trial and pursue a waiver? Today, we are joined by an exceptional group of speakers who will bring their unique insights and expertise to this very important topic. So let's dive right in. First, we'll hear from Dr. Borje Darpo, Chief Scientific Officer, Cardiac Safety at Clario. Dr. Darpo, please go ahead.
Thank you so much, and thank you for providing this opportunity to present on this very exciting topic as I see it. As you know, this area is really highly regulated. ECG evaluation has been regulated by ICH since 2005, when the first version of the E14 guidance was published. The first version presented a very conservative way of looking at ECG effects, or more specifically, a QTc effect of a new drug. In 2015, there was a major revision to the guidance, allowing us to use concentration QTc analysis. That opened two windows. One is that thorough studies, should you be required to do one, can be done more efficiently with smaller sample size. But more importantly, it opened the opportunity to apply concentration QTc analysis into your first in human trials, and thereby potentially waive the request for a thorough study. In 2022, the latest revision was published, and that revision even further facilitates using first-in-human studies to waive the request for a thorough study, and I'll come back to that. One way or another, some type of ECG and QT evaluation has to be performed for all small molecules in development. Today, we'll talk about two different pathways: the 5.1 pathway for any drug that can be given at sufficiently high doses to healthy volunteers and can be tolerated by those, and the 6.1 pathway, which is mostly oncology, for drugs that cannot be given at high doses to healthy volunteers. In 5.1, we typically evaluate the effect of a drug in healthy subjects, either in a thorough study or with the application of concentration QTc analysis based on data from the first in human trial. The 6.1 pathway is conducted in the patient population and is then not necessarily placebo controlled. In 2015, in our view, the journey towards a more efficient way of looking at potential QTc effect was initiated, and this was largely the result of the so-called IQ-CSRC study, which was conducted by industry, FDA, and Clario in collaboration. Also, by that time, 10 years down the road from the initial publication of the E14 guidance, there was, of course, extensive knowledge gained from concentration QTc analysis being applied to not only thorough studies, but also to first-in-human studies. And I think it's fair to say that today, most first-in-human studies are conducted with implementation of serial ECG monitoring paired with PK sampling. This is often done in both the single ascending dose part and the multiple ascending dose part. And since you never know definitively how the PK of your drug will look like, it makes sense to store the data and make the decision what to analyze at a stage when you know more about the project viability, but also know more about the observed PK from the first in human studies. Also, when you are required to do a third study, the needed sample size to exclude the small effect, to exclude the 10 milliseconds effect, is largely reduced by using concentration QTc analysis. So what is the difference between the traditional way we initially analyzed a therapeutic study and concentration QTc? On the left side, you see a typical data from a typical four-way crossover study, a therapeutic study, with a drug that is called vatiquinone. And we're looking at the green line is the moxifloxacin response, so the positive control, whereas the red and blue line is the placebo-corrected change from baseline of the active drug. The y-axis gives you the main parameter, the placebo-corrected or placebo-adjusted change from baseline QTc, whereas the x-axis gives the time points. So when we analyze a therapeutic study this way, we are asked to exclude an effect about 10 milliseconds at each and every time point. So in essence, this means that we're repeating a statistical test at each time point. This is, of course, a conservative way that will require relatively large sample size, typically in the range of 40 to 60 subjects. On the other hand, when we take the same data and analyze the data using concentration QTc analysis, we use all data into one model, and we then look at the effect on the same parameter, the placebo-adjusted change from baseline QTc, as a function of increasing plasma concentrations. And as you can see from the right slide, the relationship between the parameter, the QTc interval, and the plasma concentration is very shallow. The mean slope is very shallow. And we can also see that the upper bound of the confidence intervals the upper bounds are clearly below the 10 millisecond threshold. So either way, this study is negative, but the right-hand concentration QTc analysis is much more efficient. And the same study as would need 44 to 60 subjects will now need around 24 to 28 subjects in the crossover design to show the same thing, that is to exclude a small effect on the QTc interval. This is one slide from the IQ-CSRC study. And then we're looking at 9 subjects in active and only 6 on placebo. And of course, the main analysis here was based on concentration QTc analysis. And we're looking at the same type of slide with the concentrations on the x-axis and the placebo-adjusted change from baseline on the y-axis. The black line is the slope of the concentration QTc relationship, and the gray shaded area is the confidence interval. And we can see a slightly positive slope, and we can also see that at anticipated therapeutically relevant concentrations, somewhat above 200 nanograms per ml, we cannot exclude that this drug has an effect exceeding 10 milliseconds. The upper bound of the confidence interval is above 10 milliseconds. These are some items that are important to have a good control of, and I'll just walk through the 2 most important ones. In the 2022 guidance, a new concept was introduced called the high clinical exposure scenario. This was what we used to call the worst case scenario. The high clinical exposure scenario is the mean Cmax, the mean peak concentrations at steady state in the patient group that has the largest effect on clearance, be it because of a drug interaction or intrinsically, that is hepatic or renal clearance. So as an example, if we have a drug that is a 3A4 substrate, you need to look at what happens with the mean Cmax at steady state in patients also on a potent 3A4 inhibitor, such as itraconazole. When you know that, or when you can guesstimate the Cmax and steady state and those in that population, then you have defined your high clinical exposure scenario. And that is a key concept that you need to bear in mind when you decide whether to analyze the first-in-human study, and also if a waiver for the third study is at all possible. This is another slide from the IQ-CSRC study, now looking at the negative compound, DVCR. And here you can see that the slope of the concentration QTc analysis is very shallow. You can also see that the upper bound of the confidence interval, which I show here, is below 10 milliseconds. Now, what matters then, it's clear from this graph that you would like to have a narrow confidence interval. And what then affects the confidence interval? Two main parameters, I would say. One is that you're working with a site that has experience with this type of assessment and therefore knows how to handle the subjects, making sure that subjects are supinely resting at the predefined time points with no disturbing elements in the immediate surrounding. The other important component is to use a highly precise ECG methodology, because the less precise or the more variable ECG method you use, the wider confidence intervals you will see, and thereby you run the risk of exceeding the 10 millisecond threshold. So this graph outlines the main differences between a standard technique, what we call a semi-automated technique, and the technique that Clario uses, the EPQT. The upper row shows you the standard technique, by which we extract 3 ECGs, either recorded by ECG machines or extracted from Holters. From these 3, the triplicate ECGs, we analyze 3 beats from each replicate. So in summary, 9 beats at each time point. Initially, the computerized algorithm places the measurement points, the calipers, and then these measurement points are adjusted by the technician in a way that he or she is comfortable with. So we end up having measured 9 beats per time point with a method that mixes a computerized measurement and a man-made measurement. This is a very robust technology. It has been in place for more than 30 years, and we, Clario, always uses this technique when we are performing ECG analysis or patient studies, because in patients it's more commonly seen that you have T-wave morphology changes. The lower panel shows you the EPQT technique, whereby you extract 10 ECGs at each time point. All beats from these 10 ECGs are looked at by the computerized algorithm. So we're looking at basically 100 beats and categorized into high confidence or not so high confidence beats. The high confidence beats are measured on by the computerized algorithm and accepted, whereas the not so high confidence beats are looked at by the technician. But there is never any adjustment of the measurement points. Either the beat is accepted because it's within a certain threshold, or it is rejected. So we end up having measured around 100 to 120 beats per time point instead of 9. And this then translates into precision of the parameter, the change from baseline QTc. This is a slide taken from a publication that we did in collaboration with Borje some years ago. We're looking at 7 different therapeutic studies. The red lines being studies conducted with a standard semi-automated technique, and the black lines being 3 studies conducted with EPQT. The same sites were used, and these were all therapeutic studies, so positive control and active drug at 2 doses. The upper panel shows you the comparison in the crossover design studies. And we can see here that the variance or the variability using EPQT is roughly half of that with a standard technique, both for the comparison to test drug versus placebo, as well as the positive control versus placebo. And the same holds true when we look at parallel design studies. So EPQT as a method roughly halves the variability as compared to standard technique. And this, of course, matters when the task is to exclude a small effect on the QTc interval at sufficiently high concentrations. These are simulated data that we've done both internally and also and published from internal efforts, and then also in collaboration with FDA. If we assume, which is a standard assumption, that you have a small underlying effect of 3 milliseconds, and then if we use EPQT that has a typical standard deviation with change from baseline of around 6 milliseconds, you will need 9 subjects on sufficiently high doses and 6 on placebo to exclude that the drug has more than 10 milliseconds effect, like the levocetirizine graph I showed you. On the other hand, with the same underlying assumptions and with a good semi-automated standard technique that comes with a standard deviation of change from baseline of around 10 milliseconds, you would need roughly twice as many subjects. So that's why it's important to first work with a site that is familiar to this type of studies and to the ECG evaluation, and secondly, with a sufficiently precise ECG method. The 2022 revision of the ICH E14 guidance became the S7B E14 Q&A document. And the background to this was an increased interest in non-clinical assays, much promoted and spurred by FDA's interest in this called CEPA project, which was initiated by FDA. The CEPA hypothesis was that if you conducted a set of assays, non-clinical assays, and then paired that with a sub-interval of the QTc interval, the JT peak interval, you would not need to run a therapeutic study. So more non-clinical assays were introduced, as well as a new parameter on the clinical ECGs. The driver behind this was, of course, both the wish from industry and also the wish from regulators to reduce the number of standalone therapeutic studies. However, there was not much of interest from other regulators, and industry wasn't really interested in taking on board more GLP compliant non-clinical assays. So the CEPA didn't make it into the guidance, but it was a main component behind the promotion of non-clinical assays, which we will see now plays a role in how you can determine whether you can waive the request for a therapeutic study. So it used to be that, based on the 2015 revision, that if you obtained plasma concentrations twofold above the high clinical exposure scenario, so that brings that parameter back again, that if you achieve twofold higher concentrations in your first human trial and can show that at these high concentrations the study is negative, that is, you can exclude the small effect, then these data will waive the request for a therapy study. With the 2022 revision, you only need to cover the high clinical exposure, provided that you have conducted the HERG assay and the non-clinical, non-rodent in vivo study as per best practices, which are now described in the guidance. So if you do these as per best practices, you can substantially reduce the needed plasma levels in your first human trial to waive the request for a therapy study. So this is one example. The left graph shows you a typical scatter plot with a linear regression, which is very shallow. So you can see that the concentration QTc relationship is very, very shallow. And the right side shows a clearly negative concentration QTc goodness of fit plot. We can see at the very highest concentrations, the upper bound of the predicted effect is clearly below 10 milliseconds. Now, this drug has not yet established the high clinical exposure scenario. So then there are basically two options for the sponsor in case the non-clinical package does not meet best practices. Either they can repeat, as an example, the HERG, if the vivo study is negative, and then claim that these data will be sufficient, provided that you cover the high clinical exposure scenario. Or they can add additional subjects, which is rarely a good option. But this way, the S7B E14 guidance provides new opportunities to replace the therapy study and waive the request for such a study. So to summarize the revision, the 2022 revision of the S7B E14 guidance for the 5-point pathway that applies to drugs that can be given at sufficiently high concentrations. We now need to achieve the high clinical exposure scenario in terms of plasma concentrations in the first in human trial. If we then have conducted the in vitro assays as per best practices, that is sufficient, provided, of course, we can show that there is no effect on the QTc interval at these high concentrations. For the 6-1 pathway, it's a bit more difficult at this stage to understand exactly what the impact of the 2022 revision will be. It used to be the case, and it's still the case, that these studies are conducted in patients, typically not with placebo. So we're looking at the change from baseline QTc, either with a by time point analysis or by using concentration QTc analysis. And if we, by looking at the highest recommended dose, can conclude that an effect exceeding 10 milliseconds can be excluded. So the upper bound, again, needs to be below 10 milliseconds. It used to be the case, and it still is the case, that the label will say that this drug is unlikely to have a mean effect larger than 20 milliseconds, which you can see under the current column. If then non-clinical assays are performed as per best practices, furthermore with the addition of a sensitivity criteria for the in vivo study, then the label may say that this drug has a low likelihood of pro-rhythmic effects based on delayed repolarization. We haven't yet seen one example of this revised label, but it may be. And one reason could be, of course, that most sponsors perhaps do not see a huge difference between the current statement of unlikely to cause an effect as large as 20 milliseconds and the revised statement in the label, proposed statement in the label. So what is the likely impact of the 2022 revision? Well, first of all, I think importantly, it will further increase the proportion of first-in-human studies to replace the request for a third study. Whereas I do believe that it's more difficult to predict what the effect will be on the 6.1 pathway. Most of the studies I see today are still conducted in-patient studies and are looking at the change from baseline QTc, even though we haven't seen, of course, what the label will be based on how the non-clinical package was conducted. Non-clinically, and I think this is important, most sponsors should liaise and will liaise with their laboratories, making sure that the HERG assay meets best practices and the in vivo non-rodent study does the same. So my conclusions based on the 2015 and the 2022 revision of the SMB E14 guidance is, first of all, that applying concentration QTc analysis on the first-in-human trial is a fully accepted way to exclude that the drug has a relevant effect on the QTc interval and thereby waiving the request for a third study. I think the revision of the guidance in 2022 will further promote the replacing the third studies with first-in-human trials. I think it's worthwhile to emphasize that it's critically important for sponsors to liaise with the non-clinical laboratories. I also believe that the third study will not completely disappear and will remain a viable option for those sponsors that didn't do much of this serial ECG monitoring in their first-in trial. I think it's also fair to say that QTc evaluation over the last 10 to 15 years have been made much more efficient by the combination, of course, with many more sites having experience, but also using concentration QTc analysis and the involvement of highly precise ECG methods. Thank you so much.
Thank you, Dr. Darpo, for that great presentation. Next, we'll be hearing from Justin Couture, Director of Clinical Operations at Dr. Vince Clinical Research. Justin, the floor is yours.
All right. Thanks, Janelle, and thanks everyone for listening to me talk a little bit about the clinical considerations for TQT trials. Let's go to the first slide. So Dr. Darpo gave a great presentation about the data analysis. What I'm talking about more is the initial collection of the volunteer cardiac data. So it all starts with volunteer education, letting them know from screening to admission to treatment what to expect from a TQT trial. So what that data means to the success of the trial, what to expect from a more pragmatic sense in terms of restrictions on electronics, anything that can interfere with our cardiac data quality, and the amount of time they'll be spent just honestly just laying in bed, laying still, staying awake. And what we do at Dr. Fitz Clinical Research is we do educate them on that, and we post the exact times up in each suite to let the volunteers know during these time periods, you are required to stay in bed or to avoid use of electronics. So that's key to just set a precedent for the expectations of that trial. Next slide. Another key aspect, the data analysis on the back end is only as good as the data that we provide from the site. So as Dr. Darpo alluded to, skin preparation, appropriate placement of the electrodes on the volunteer's chest, that stuff's critical to ensure that the data is of the highest quality. So that means, using electrodes, skin prep pads to ensure there's no oils or lotions in place that would interfere with the placement of those electrodes, shaving if necessary, using anatomical landmarks, counting intercostal spaces to ensure that you're placing each sticker on their chest in the appropriate location. And then for our specific monitors here, ensuring that we have a bag for them to place the physical monitor in so that there's no tension on those leads, which just reduces stress on the cables and the potential for those leads to come unhooked and affect that data. Next slide. So here at DVCR, we have what's called a central monitoring station, a Hill-Rom Surveyor station. So we have 48 channels. What that means is that we can monitor up to 48 volunteers simultaneously from one central location, as you can see here in the photo. So from a safety perspective, if we're monitoring cardiac telemetry, that means we have one of our advanced cardiovascular life support certified staff at that location monitoring in real time. What it means for Holter monitoring is that we just have a sort of a one-stop shop to view everybody's cardiac rhythm simultaneously, rather than having to go from bed to view each volunteer. It is a Wi-Fi based system, so all of it's automatically transmitted and saved on site as it's collected in real time, which is a huge benefit to us here at DVCR. Next slide. So this is an image of one of our S4 mobile monitors. They're very small, about 6 inches. They're lightweight. They run off lithium batteries, so they can run for 24 hours plus if we're just monitoring cardiac data. They are all 12 lead as a standard, and like I said, they communicate with that central station via Wi-Fi. And one of the perks of this system is we have certified with Clario as a Clario certified site. What that means is that we utilize this system to qualify as what's called site champions, which basically means that two of our staff, a minimum of 2 of our staff, went through a rigorous training program and collected 3 8-hour Holter monitoring sessions each, run just like a standard TQT trial, so paired up with PK, multiple time points for extraction throughout the day. We collected all that data, which we renew every 2 years, and we utilize this system, so we're able to archive this and submit it directly through Clario's portal online. One of the benefits of this being a 12-lead system is one system allows us to run safety ECGs, as well as collect Holter data, as well as monitor cardiac telemetry. So really, it's sort of one system handles all 3 different aspects, which is important when you're running Holter trials because, as we said, you have to make sure that lead placement is precise, and you're not sacrificing that quality, whether you're using it for Holters or whether you're using it for safety ECGs. Next slide. So the operational benefits, I touched on some of these. We're able to use that system for everything. That what I just mentioned, the one set of electrodes, that's huge because you can't compromise in a TQT study on the quality of that cardiac data, and we don't have to because of this system. It also allows one central location for any alerts that can affect the quality. So if the battery is running low in the device, if the V2 lead is off, if there's any sort of alert, a significant artifact, things that we're trying to mitigate in the clinic, but that sometimes you can only see by looking at the screen. This allows us to go in and say, okay, I see all 20 volunteers who are on Holter monitoring, and I can quickly scan before my extractions and say, the leads all look clear, there's no lead missing, there's no limb reversals. We do still ensure that our staff assigned to extraction time points physically verify all the leads on their chest before each extraction time point. And that really helps with missing some of the more subtle issues, like if a lead is reversed, a limb lead is reversed, something like that. But that central location really does assist us with getting a very quick snapshot of how the quality looks across the board for an entire group. Next slide. So once we've completed our Holter monitoring session, as I said, it's Wi-Fi based. So another benefit of this system is that it doesn't use SD cards or Bluetooth, anything like that. It's all from our Wi-Fi. So we're able to say we completed 24 hours of Holter monitoring for these volunteers. We're able to archive that data directly from this Surveyor system, and that saves it to our on-site hard drive and then a cloud-based backup indefinitely. So we can archive that data to ensure we always have it. And then without getting into the details, because it's not overly exciting, but then it's a lot of file management, a lot of naming conventions that make sense, ensuring that you have the correct file for each volunteer, QCing to ensure all that data is present. What that looks like typically on our end as a site collecting the data is verifying the start and end time of the Holter are accurate and present, to ensure that we have that full 24, 25 hour, whatever the time frame is, that full amount of data. Next slide. And then the data transfer process from site to Clario is very simple. And as I said, we went through that in the site champion process. So once those files are saved and archived and named appropriately and organized, we convert all of those files to a zip file. And then every time we have a new trial that we're working with on Clario, we create a specific site where we assign roles to everybody on our team, and we submit like a 2-hour typically test Holter just to ensure that the data is transferring correctly. So once this data is archived, we'll convert them to zip files, and then we can upload them directly through the web into Clario's portal that was created just for that study. And that's where Clario will do their review and then their analysis depending on the requirements of the trial. Thank you for your time, and I appreciate you hearing me out.
Thank you, Justin. Up next is Michael Hirschman, Lead Project Manager at Clario. Michael, over to you.
All right, thanks, Janelle. As Janelle mentioned, my name is Mike Hirschman, and I am a lead consulting project manager here at Clario. I've been with the company for 10 years, and I oversee a team of project managers that specialize in the end of study statistical analysis, cardiac safety report generation, and regulatory submission support. So now that we've heard from Dr. Darpo and Justin, I would like to discuss how this works operationally at Clario, review our standard timelines, and detail the support you will receive from Clario for regulatory submissions. So on screen right now is an overview of the operational workflow showing Clario's involvement from study start through study submission. So at the initiation of the study, you'll be assigned a core project manager, and they're going to coordinate the clinical operations of the study. You'll also be assigned a data manager and a consulting project manager who oversees all of the post-database lock activities. And this graphic shows each functional role's involvement and the major Clario milestones throughout the project. Next slide, please. And here is a more in-depth look at the analysis activities and standard timelines. And just a quick note that the activities in blue indicate where sponsor input is required for a certain task. So starting on the left at step 1, Clario is going to create a draft SAP that's going to outline the ECG objectives, the endpoints, methodologies, analysis populations, planned analysis, and the actual TFL outputs. So that SAP is going to be reviewed by the sponsor. revised if required, and mutually approved by the sponsor and Clario. We aim to have this step completed at least 4 weeks prior to last subject last visit, but in practice, it often happens much earlier in the process, often while the clinical conduct is happening. Once the SAP is approved and both the clinical and the Clario databases are locked, your consulting project manager is going to reach out and request for you to share required CDIS data sets for our analysis. The requirements for each study are going to be detailed in a data import agreement, and the data sets are described in the following slide. Moving on to step 3, after we receive the needed data sets, the Clario programming and biostats team are going to develop and validate our analysis data sets, perform and validate the analysis as outlined in the SAP, and then the results are interpreted and presented in the expert cardiac safety report. This report is typically provided 8 to 10 weeks after receiving the data sets detailed in step 2. From this point, the report's reviewed, revised if required, and then mutually approved. And then to finish up in step 4, following the report finalization, we're going to provide an electronic submission package, which I'm going to discuss in more detail in just a moment. Next slide, please. All right. I'm going to briefly touch on the dataset requirements. So as I mentioned, the requirements for each study are going to be laid out in a data import agreement. However, the 3 data sets here are the standard CDIS data sets that we are going to need to conduct a concentration QTc analysis. First is the SDTM-EG data set. That's going to contain the Clario overread ECG data, the ADAM ADSL, and that's going to contain the subject demography and treatment information. And lastly, the ADAM ADPC, which is going to contain the time point level PK information. And just as a quick note, we can't begin our analysis until all of these data sets are available. The next slide, please. All right, moving on to the electronic submission package. This is typically one of our final deliverables. So following the cardiac report approval, we're going to provide a package of supporting materials that can be incorporated into an overall study submission package. What we are going to include is the Clario created analysis data sets, the analysis data reviewers guide or ADRG, define XML, dataset specifications, the SAS programs for the Clario datasets and the TFL outputs, Pinnacle 21 validation reports, and lastly, all tables, figures, and listings as RTF files for incorporation into CSR. Next slide, please. And finally, as part of a submission, the FDA recommends completing and including the QT evaluation report submission checklist. At the conclusion of the study, we're going to provide this checklist with as much information completed as possible. We're going to highlight and offer guidance on the items that must be completed by the sponsor. So what we are going to include is everything that I described in the electronic submission package. We're also going to provide the SAP, the final cardiac safety report, details on the ECG collection and analysis, and the annotated ECG waveform files, which are also known as the FDA XMLs. The sponsor is going to be responsible for providing the investigator brochure, the highlights of clinical pharmacology document, integrated categorical analysis, and narrative summaries and case report forms. So to summarize, Clario is going to provide everything we can to support your submission. And following the expected deliveries, the Clario team is going to remain available for ongoing submission support and guidance. All right, thanks. Back to you, Janelle.
Thank you all. Those were excellent presentations. We've received many great questions. Thank you to everyone who has submitted thus far. Feel free to continue adding questions as you think of them, and I will do my best to combine any duplicates. We'll address as many as possible with the time we have and follow up after the webinar for any questions we don't get to. All right, going back towards earlier, the high clinical exposure scenario is not known when the first in-human study is conducted. How would we then know that analysis of the ECG data from the study can waive the request for a TQT study?
Well, the simple answer is that you cannot know that. you first need to characterize the PK profile in patients to have a better understanding. Sometimes you can estimate that the drug is a 3A4 substrate, as an example, or a 2D6 substrate, and you would have an understanding, and you may have an understanding of how much a potent inhibitor would increase the Cmax at steady state. In most cases, you can't really make a definitive conclusion about the high clinical exposure scenario, and therefore this justifies that you store the waveforms until you know a bit more about the patient PK profile. Then when you know not exactly everything, but when you have a fair idea of the patient profile, then you can make an assessment whether it makes sense to analyze the first in human data, the ECGs and PK data from the first in human data.
Thank you. A follow-up on that, a double negative non-clinical package performed as per best practices seems important. How much deviations from these practices will be accepted by regulators?
Well, that's a very interesting question. And the best way to know that for sure is by accumulating experience with interactions with FDA's IRT team. Dr. Vicente from IRT gave a presentation almost a year ago before leaving FDA, where he shared some examples. And the one that struck me was a Herg assay conducted at room temperature. As you know, the best practices requires near physiological temperature. On the other hand, the safety margin for Herg was like 800-fold. So then it really doesn't matter, because the examples we've seen with huge differences between room temperature and near physiological temperature is not in the range of 100-folds. It's possibly 10-fold. So in that case, the Herg assay was still accepted as meeting best practices, because the safety margin between the Herg IC50 and the free concentration seen in humans at the Cmax steady state was so large that justified accepting the Herg assay. So it will be a case-by-case scenario. And of course, the higher safety margins you have, both for your in vivo studies and the Herg assays, the easier the discussion will go with the regulators, in my view.
Thank you, Dr. Darpo. Next question, how long are volunteers typically monitored? How are longer duration monitoring periods handled differently from an operations perspective?
Yeah, I can speak to that. Typically, in my experience, 24 hours is relatively standard. I have seen Holter monitoring sessions up to 96 hours. Once you get, from an operational perspective, it ties back into what I mentioned earlier in the presentation. A lot of it is just education. Even 24 hours feels much longer to these volunteers when they're spending the majority of their time off of their phones, off of electronics, not sleeping, and just laying still for usually 15 minutes at a time, if not more. So once you get over 48 hours of Holter monitoring, there's a couple key considerations. Education is obviously the first thing, no matter the duration. Another is using skin markers to mark where those electrodes were placed. That allows us to have that reproducibility on site, so that Dr. Darpo, when they're analyzing this data with their systems, repatching the volunteer, if a lead comes off, or if we allow showers, which we typically try to, once you get over that 48-hour threshold, it's important. For multiple reasons, the volunteers would like a shower. We typically work with the client to work that into the protocol, and we watch that pretty strictly to make sure it's not 2 hours of a shower time, and that we're able to replace those electrodes in the same spot. So those are some of the key considerations to make sure that we're still getting that quality data from the volunteers.
And next question, what lessons have you learned using a central monitoring station for TQT and EPQT trials?
Yeah, so I mean, I think the benefits far outweigh anything negative with the system. It's important. The biggest challenge is really, as I said, there's 48 channels of monitoring, right? And each channel can monitor a specific volunteer. So if channel 1 is monitoring volunteer ABC, because it's a central station located away from the volunteers, it's really, really important to make sure that channel 1, that device is on subject ABC, because you can't see the volunteer when you're at that station. It sounds a little complicated. It's always important to make sure you have the right patient and the right data. What we do to mitigate that here on site, we have an online data collection system, a computerized data collection system. So we utilize that, a barcode system, to ensure that when the volunteer is hooked up, we scan the device, which puts a barcode in there, and then we start monitoring, we scan a corresponding barcode. So those two barcodes have to match for us to know that we're monitoring the correct volunteer on each channel. Other than that, it's all benefits. And like I said, that's a key no matter how you monitor, but that's what we do to mitigate that challenge.
All right, thank you. There's a lot of questions coming in. Let's go with this next one. Is there a way to expedite the time between DBL and when the results draft cardiac report is available?
Yeah, I can jump in on that one, Janelle. It's a common question that we get. Everyone's interested in getting the result as quickly as possible. So what we can do, and this will be offered by your project manager at Clario, is move that SAP creation up a little bit so it happens earlier in the process. And then we'll also request for dummy data sets. So it's those 3 data sets that I described, but with randomized data in there, not the actual study data. And the Clario team's going to take the finalized SAP and then those dummy data sets and move a good deal of the programming work prior to database lock. And by doing that, once database lock occurs and the final unblinded data sets are available, we can just refresh our analysis with that. And using that method, we can generally shave 2 to 3 weeks off the overall timelines.
Thank you. What if a sponsor is not sure their FIH study will progress to submission of a TQT waiver request?
Sure. So I think this, loops back to what Dr. Darpo said for his first question. It's always valuable for these studies to collect the data, and then we can store it here at Clario. So it moves through our entire standard process. It's collected at the site. It's sent to Clario. We go through our query process to make sure that, all the information needed for analysis is there. And then we pause at that point until we're given the green light, if other study endpoints are met. And by doing that, we have the data cleaned and ready to go to move right into the overread analysis and subsequent statistical analysis.
I think we have time for one more question. Does Clario write the full CSR?
Sure. So I can jump in on that as well. Clario doesn't write the full CSR. We provide a standalone cardiac safety report. And that cardiac safety report is often either incorporated into the CSR or included as an appendix to the CSR.
All right. Well, thank you so much to all 3 of you for sharing your time and expertise today. Thank you to the audience. If we didn't get to your question during the session, we'll be sure to follow up with you afterward. As a reminder, a recording of this session will be available on demand within 24 hours. Have a great rest of your day, and we will see you next time.